You're actually never really alone. I mean, right now, inside your own body, bacterial cells outnumber your human cells. It's kind of wild. Today, we're going to dive into this unseen world to meet these tiny but incredibly powerful organisms that have been on this planet way, way longer than we have.
So let me ask you this. What's the real difference between something being clean and something being sterile? In our everyday lives, clean is usually good enough. But in a place like a hospital, it's a completely different game. We're going to find out why that difference can literally be a matter of life and death.
All right, so our story starts with the prokaryotes. These guys are some of the oldest life forms on Earth. We're going to be focusing on bacteria in particular, a group of single-celled organisms that are just absolute masters of survival.
Okay, with trillions upon trillions of bacteria all around us, how in the world do scientists tell them apart? Well, it's kind of like a microscopic detective story, and we're about to uncover the exact clues they use to solve these tiny mysteries.
The very first clue in our detective story is always the bacterium's shape. It's their calling card. And they basically come in three main flavors. You've got the round ones, called coccus, the rod-shaped ones, called bacillus, and the twisty, spiral-shaped ones, spirillum. And there's a cool scientific term for this, the study of a bacterium's shape and arrangement. It's called morphology. And this is really the foundation of microbiology. It's step one in identifying these tiny organisms and figuring out what they're up to in the world.
But wait, it's not just about their individual shapes. How they hang out together is another huge clue. Some of them form these long chains, which we call streptococcus. Others bunch up together like a cluster of grapes. That's staphylococcus.
So the next clue is all about how they move. Are they swimmers? Some bacteria are modal, and they use these incredible whip-like tails called flagella to propel themselves around. They're like little outboard motors.
Now let's talk about how bacteria feel about oxygen. It's fascinating. Some, just like us, are called obligate aerobes. They absolutely have to have it to live. But then you have obligate anaerobes, and for them, oxygen is totally toxic. And then there are the super flexible ones, the facultative anaerobes, who are just like, with or without oxygen, I'm good.
So we've seen how to identify bacteria from the outside. But what's going on inside? Let's take a little tour and look at the amazing internal machinery that makes these organisms so unbelievably resilient.
Okay, so this is the basic blueprint. But look, it's way different from our cells. Their DNA isn't all neatly packed away in a nucleus. Nope, it's just kind of floating around in the cytoplasm with everything else they need to survive and, do their thing.
The first line of defense for a bacterium is its cell wall. You can think of it like a rigid suit of armor. It's made of this really unique stuff called peptidoglycan, and it gives the cell incredible strength and protection.
And if that suit of armor isn't enough, some bacteria have an extra layer of protection, a slimy capsule. This gooey outer coat helps them do things like stick to surfaces. Yep, like plaque on your teeth, and it keeps them from drying out.
Hey, remember those flagella we talked about, the little propellers? Well, there's more to that story. The number and arrangement of these flagella can be really different from one bacterium to the next, allowing for all kinds of movement and giving us yet another clue to their identity.
We've seen their armor, we've seen their propellers, but now we're going to get into their ultimate superpower. A survival tactic so incredible, it honestly sounds like something straight out of a science fiction movie. This superpower is called spore formation. Now, this isn't about making more bacteria. This is all about survival. When things get really tough, no food, too hot, whatever, the bacterium basically packs up its essential DNA into a tiny, indestructible time capsule and goes into a deep, deep hibernation.
So, have a look here. You can see a real-life anthrax spore. It's a tiny fortress built for one purpose, to protect the bacterium's genetic code. The original cell actually sacrifices itself, leaving behind this incredibly tough spore, just waiting for conditions to get better again. And when I say tough, I mean tough. These spores are the ultimate survivors. They can lie dormant for years, decades, maybe even centuries, withstanding boiling temperatures, harsh chemicals, and total dehydration. They are, for all practical purposes, nearly indestructible. So, we've learned about these amazing, almost unbelievable survival tactics. But why does this all matter? Well, it turns out that understanding these tiny organisms is absolutely critical for keeping us safe in the real world. Let's see how.
Remember our question from the beginning about the difference between clean and sterile? Well, here's where it all comes together. If you have to sterilize surgical instruments, how can you be absolutely 100% sure you've killed every single microbe, including those nearly indestructible spores?
The solution is it's ingenious. Scientists basically use the bacteria's own superpower against them. They take harmless bacteria that are really good at forming these tough spores, and they stick them inside a sterilizer. If the process is powerful enough to kill these ultimate survivors, then you can be absolutely confident that everything else has been wiped out too.
So the next time you're in a hospital, just think about that unseen battle that's being fought every single day. It's a battle fought with science, with skill, and with a really deep understanding of the incredible, invisible world of bacteria.
Honestly, the world of bacteria is a powerful reminder that there's a whole universe of life that's completely invisible to the naked eye. It really makes you wonder, what other secrets are just waiting to be discovered right under our noses? Thanks for joining me on this little microscopic adventure.